Thanks.
Oh, can't hear you.
Yeah, your audio's off. Um, you're not mute, but we can't hear you.
I don't think he knows.
He might not be able to hear you.
Oh,
Exactly.
maybe not. Let me wonder if I could text him. If he sees the chat.
You can always stop his share.
Oh, good point. Thank you, Todd. That's a uh maybe I can't though. Oops, I don't want to share that. I don't want to screen share. Stop share. There we go. Um how to
There, did I do it?
No, we just saw your screen.
Okay, well, he'll see it too.
I saw what you texted.
Okay.
That's it. I'll stop his sharing and then maybe he'll notice. Hey, Jiyao, we're not hearing you.
Okay, it looks like microphone is not working, so I'm gonna just try it. Can you hear me now?
It's a little quiet.
Okay, let me adjust my.
Yeah.
Okay, it's better, right? Okay,
Yes.
good.
And I think you should be able to share now.
Okay.
Actually, yeah, perfect.
Let's see.
Yes. Great.
Okay. I may come back a little bit. So iCn3D can be used in different environments. I'm gonna focus on web browser, which has a shareable URL feature. It can be used with scripts to process many structures. It can be used in Jupyter Notebook. It's available in NPM. It can be used in virtual reality and augmented reality. It can be used with 3D printing. So first step is to load a 3D structure, either by IDs, could be PDB or AlphaFold IDs, or you can import a PDB file or MMC files. So once you load a structure, you can rotate with your mouse, left mouse. You can zoom in with the middle mouse and translate with right mouse. But in the iPad, you could use other features as well. Oops. So iCn3D show 3D, 2D, and 1D together in a synchronized way. And here in the analysis menu, one of the body is called 2D diagram or 2D cartoon, which shows 2D. And the sequence and annotations shows 1D sequence among all those annotations in tracks. And defined sets show the selections, which is crucial when you want to do analysis. So you can select a subset and apply different style or color. So one thing about iCn3D is you might select a subset. And so this toggle will toggle between a subset or all items. So for example, if you accidentally select a residue and you apply color or style, it might not work as you expect. You might expect the whole structure. So you can click the toggle to switch between these two. So in 3D, you can hold your Alt key and click on the 3D structure. That's a way to select on 3D. By default, you select residue, but you could select a chain of helix or strands. And you could select on the defined sets or in the 2D diagram, the nodes. You could also drag on the sequence when you select the detail view. You can drag on the sequence to select a subset. Once you select a subset, this toggle becomes orange. It mentions the selection. Now you are working on your selection. All your following style, color, etc. will be only work on your selection. So pay attention to this position, whether it's a whole structure or just a subset. So once you select a subset, you could just view this subset. You can click the view selection, will show me this subset. And the 2D diagram will also only show this part, but the sequence will show just this chain. Once you select a subset, then your style or color will only apply to your selection and color as well. So, for example, in this case, show the second structure sheet in green and helix in red, I think. Yeah. Okay, so this is just a random custom view. So once you finish your work, you can save. There are two ways. One way, more generic, is you can save iCn3D PNG image. So you can go to File, Save File, iCn3D PNG image. You could save both the image and HTML. The HTML actually, it's a kind of a gallery. You can like combine multiple such files to form a gallery. So basically, if you open HTML, you can click on the image and go to your saved view. And the PNG image can also be loaded back into iCn3D. If your input is ID, not a PDB file, you could save your work in a shareable link. So this is the full command, and this is the abbreviated command as highlighted here. So basically, we convert all your commands into a shareable link because each step of your operation has a command. Okay, so now I'm going to talk about some of the more advanced features about iCn3D. You don't need to follow along, but you can just, remember there are such features. So the first feature is about annotations. Remember, you can open the sequence and annotation windows, and there are different kinds of annotations like conserved domains, 3D domains, clean bar, snips, binding sites, transmembrane. Recently, we added IG domain detection. In this case, it shows automatically the IG domains in your structure. So each IG domain will have the strands color differently. So in this case, it has 3 IG domains and also show up here. And the IG domain is through alignment with a TM score 0.68. So this chain has 1 IG domain, this region, and we have 3 chains here. So there are 3 IG domains. I'm going to talk more about IG domains in the antibody engineering hackathon. So all the annotations can be exported. There are different ways. First, you can use Node.js script. in this URL, I'm gonna upload this PowerPoint into one of the Slack channels, so you can follow these scripts. So the script is located in the iCn3D GitHub page under iCn3D node. So you can get the annotations with Node.js script. You can also use Python script. I think it's this batch export panel.py. So this example script will export the annotations. Basically, it's similar to your web UI. We'll export all these annotations. So let me talk a little bit more about IG domain. So once you load a structure into iCn3D, once you click the IG domain annotation, it will automatically detect the IG domain. This is 1CD8, and it's an IGV domain. It's labeled here IGV because it has its own template. We use template to match the IG domains. So that actually the TM score is perfect match. It's 1. And it defines where the IG domain is located. So the first residue is not a part of the IG domain. It's N terminal. And then each IG domain from A strand to G strand are labeled with different colors. So this is a little bit complicated slide. So basically, you can compare IG domains in different structures using the IG reference numbers. So, for example, the left part shows the IG interactions, and the middle part also shows IG interactions in another protein, and they have different residue numbers. So in order to compare them, you can use a reference number, which, the same reference number indicates the same location. So with the reference number, you can compare the different interactions in these two interactions. So you can remove the common interactions. Also, I think actually it's last month, we add a new feature to iCn3D ligand protein interactions. So previously, for the glyvac interacting with protein, we show the 2D interaction network. It shows ligand as one residue, and all the interacting residues are showing up in another line. Now we added a 2D interaction for one ligand or one residue. So basically, we show the atom details of the ligand, like here, and then show the interacting residues. And the color of the interactions are showing here. So the green is the hydrogen bond, and the red is the cation pi interaction. And you can customly move these residues to achieve a bad display. You can play along with that. So I think I talked about the mutation analysis before. So basically, in the analysis menu, you can click mutation, then specify which structure, which chain, and the position of the residue, and what will be the mutant residue. So once you define this, then you can, this works for PDB ID or AlphaFold ID, and then you can show in the current page or in the new page, show the interactions. Here shows the mutation of the asparagine to tyrosine in SARS-CoV-2. spike protein. By the way, when you do the mutation analysis, by default, both the wild type and the mutation are showing up. You can click the letter A to shift between these two structures. iCn3D can also show delphi potential with one simple click. So once you load your structure, you did your selection, you can either show the potential on surface, which is the protein part, or show the potential as an equipotential map, which is shown for the ligand part here. iCn3D has lots of alignment feature. You can start with the align file. In the file menu, there's align option. Or you could load structures first, then do the realign. And the alignment has the protein complex alignment via VAS plus, and or chain alignment via VAS or TM align. And the align could be based on structure, sequence, or residue mapping, basically user-defined alignment. I'm going to show some of the alignment, some examples. So if you go to File, Search Similar, and you can click VAS plus, and you can input your structure, for example, 1HHO, and click VAS plus. It will find similar. It will find a similar alignment. Basically, it's an alignment for the complex. So, for example, if you expand this one, these two are similar to the starting. I think, sorry, in this case, the PDB ID is 6M0J. It's a little messed up here. So basically, your query structure, your match structure with similar alignment. And you can also go to NCBI vast alignment. So you specify your PDB ID and chain name, and then you can click vast to show similar chains in this page. And then you can view 3D alignment in iCn3D. And you can also do a vast search. So for example, you load a structure, you can just submit into vast search. So once you click submit, there's an intermediate page, then you start the vast calculation. And then, so the final alignment will look like this. If so, for example, you start with align multiple chains, you specify the chains and align with TM align or vast. Then the 3D structure will be aligned. Also, the sequence will be aligned according to the structure alignment. So the realignment feature, for example, I load three PDB files into iCn3D, and then I click realign by structure alignment. And then these three structures will be aligned with a TM score 0.98 in this case. And the pairwise sequence alignment will also show up. And here just show one example in the virtual reality. So this is generated from MetaQuest 2. So you can go to Meta browser in the VR set and then input the iCn3D URL and your PDB ID, and then you can enter the VR view. And also there are menus in the VR mode. So basically, this will be very interesting to students for teaching purpose. And there's a VR tutorial in this URL. Okay, I think I will stop here and I can answer questions.
Thanks. I think we were wondering. Oh, Keith, go ahead.
Hi, Jiyao. Long time no see. I do enjoy
Yeah.
your presentations, and I'm really excited about iCn3D. I was hoping to take a virtual reality class last semester, but it was canceled, so I'm taking it this semester. And I have no idea what the capabilities are. Am I going to be able to, with my VR headset, walk around and reposition and expand and rotate the molecules such that it looks like I'm almost in the molecules?
Yes or no. So basically, once you enter the VR, I actually just did a VR mode, VR demo this morning. So once you enter VR mode, you are actually in the middle of the protein. So the protein is like a world, protein world, and it is a static. And you can navigate in the protein world in different directions, zoom in and out. Basically, you are moving, but the protein is not moving. It's like huge mountains. And so you can going backward and forward in different directions. So you can go to, for example, if you're interested in ligand, you can point your line to ligand and go forward, get closer, or go backward. But you cannot rotate the protein. That's one limitation of this current library. And so you can select residues. You can do simple analysis like interaction analysis, and you can change style, color, etc. So yeah. So, but comparing to nano, it's still limited. In nano, you can rotate the structure. You can have multiple users, work on the same structure. So that's a commercial software, but I think iCn3D is good enough for users to view the protein or show simple interactions. Yeah.
Thank you so much. I'm excited that this semester is going to bring forth that capability.
But that's our goal in the future when we are using 3.js library, and we depend on their capability. Yeah.
Dylan.
Yeah, is it possible? I didn't realize you could do the ligand protein interactions. Is it possible to load ligands from PubChem?
Yes. You can load PubChem compound through the PubChem CID or compound name. I think we recently added you can input
compound name
or smiles inchy as well. So
smiles. Okay.
If you go to iCn3D file retrieved by ID, and the PubChem part is PubChem CID or name or smiles. Yeah.
Okay.
Jiyao, couldn't you also go to PubChem, find the thing you're looking for, and isn't there a link that'll open it in iCn3D?
PubChem has its own 3D viewer. So right now, they maybe they would prefer a very light version of 3D viewer, so they didn't link to iCn3D. Yeah.
Dylan, I think you had a question about coding too.
Yeah, I started playing with that, and I could never actually get the iCn3D Python library installed. I couldn't find it, using pip3 or pip or conda and I wasn't sure, do I need to start with the GitHub repository and get that for the Python library?
Let's see. So iCn3D actually it's not in so
not in any of the repos. Okay.
For the Python part, we only have the Jupyter notebook version. But
Okay.
with the I don't think we are in pip library, but we are in the npm library.
Okay. Okay.
I feel like every time I hear you, I learned something new that I didn't exist in iCn3D.
Right. So I usually highlight the new ones. Yeah.
Yeah.
So
Well,
for
that
example,
was,
the ligand part was done through the ISMB meeting in July. So that was really, yeah.
yeah, I was there, and I mean, I was virtually there, and somehow I missed that. But no, that looks nice. That looks very powerful.
That's actually after the meeting. We did a code phone with Bosk. So it's after the meeting. Yeah.
I was wondering, so I think I'll have to try some of the vast options that I missed noticing before. But I was wondering if you had a structure, right? If
Yeah.
there's a way that you can use the search for similar structures to find a complex that might contain that structure. So like say you have a molecule like hemoglobin, and you want to find out if there's a complex where an antibody is binding to the hemoglobin. Can you use the hemoglobin and VAST to find structures of hemoglobin that might have an antibody bound to it?
Yeah. So yeah, I think you can do that through VAST. Yeah.
Okay. I will try that. That seems like an easier way than some of the other ways I've tried.
Yeah, that sounds good.
So it was all pretty quick, and I know some people are still, iCn3D is still new to a lot of people. Do other people have questions about it? Or is it just kind of all overwhelming? Erica.
Hi, I've been using the iCn3D with my students a lot, and maybe you could help me. Sometimes, one of the things we use with it a lot are looking and aligning sequence to the structure. And sometimes when they're pulling an accession number, they're pulling the nucleotide instead of the protein accession number. And so when they do the alignment, it's not aligning. And what I'm wondering is if there could be like a warning box or something like that that would maybe pop up and say, hey, you're trying to align, this looks like nucleotides or something like that. Does that make sense?
So I think you are more talking about the BLAST page, right? Are you coming from BLAST or you are working in iCn3D?
I'm using iCn3D, and what we do is the align and then the sequence to structure. And then there's an accession number that we put in there.
Okay. Yes. That's a good point. Yeah, so there you need to find for your input ID, you need to find a template, right? And your input might be a nucleotide, right? Yeah, that's good. That's good point. Yeah.
I just wonder because sometimes it kind of stops them and they're like, and
right,
just
yeah.
So
yeah, so it can easily tell whether it's a nucleotide sequence or protein sequence. Yeah, so that's a good idea. So we can pop up. So you should convert that into a protein accession. Yeah. Thank you.
Yeah, that's good because like if you were to do that at a BLAST page at NCBI, it would tell you.
Yeah.
But iCn3D does not. Of course, my students don't actually read it when it tells it to them. But that's another issue. I don't think you can make a warning that'll help with that. Are there more questions? I'm sure you guys must have questions or things you want better explained, or maybe Maybe can you tell us what a Delphi potential is? I know it sounds clueless, but
Yeah. So delta potential basically the electrostatic interaction is one of the, I would say, major interactions between proteins. Like usually there's van der Waals interactions. Basically, it's by contact, which is a weak interaction. Electrostatic is stronger than that, and hydrogen bond is certainly is very strong. It's almost like a bond. So I would say usually especially for example nucleotides, they are highly negatively charged, so they have negative potential, right? And if your protein has on the surface of your protein has a patch of uh, positive charge residues, it's very likely that it might interact with the nucleotides if they are getting close. So, so of course you can tell what are the residue, which residues are positive, but sometimes there's a negative and a positive together, so you have no clue whether it's the final potential is positive or negative. So with the potential, you can have a better understanding whether the final, the sum of all the contribution is more negative or more positive. Yeah. So for protein-protein interaction, I think that's very crucial.
okay. There was another question that came up that I thought about. This is completely different, but when we look at when we're looking at interactions between I don't know, an antibody and a protein, right? And we go to the analysis menu and we pick interactions so that we can see them. We can get a table of the interactions, which is great, right? The kinds of bonds and the interactions. We were wondering if there's a way to print those tables.
Interactions, let me show you.
Yeah, that'd be great. Thanks.
Yeah. So, okay. Can you still see my browser?
Mm-hmm.
Mm-hmm.
Okay, good. So, so for example, I use the and I want to see the interactions between this ligand and my called ligand. And I want to show the interactions between the ligand and maybe everything else. So one of the way is to show the details in the interaction in tables, but sort them based on set one. So here it actually shows the ligand has 5 hydrogen bonds and 30 contacts and 1 pi cation, 4 pi stacking, and then the details are listed in the table. So this is one way to show the detail of the interaction. I mean, I show the two interaction network, which is like this.
Right, but back to that table. Can we print that table?
Yeah, the table, you can click this one.
Oh.
Save, yeah. So save as HTML file.
Okay.
So once you click it, it's
yeah, okay.
formatted HTML file. Yeah. So yeah, but in the in this diagram, in this SVG viewer, you can also output the interaction in JSON format. So basically, let me see. Basically, it defines all the nodes and the links are the interactions, like source and target, and so you need to like translate. For example, yeah, the color shows like color red is hydrogen bond, but the information is here, but it's more indirect. Yeah.
Okay.
And what else? Let me see.
It was mostly the table I was wondering about.
Yeah. So all the pop-ups have this save feature.
Yeah.
Even this one you can save as HTML. So like this.
Mhm.
And so for example, this one you can also, I believe you can also save it.
That could be very useful for people that want to make illustrations for publication.
Yes. Yes. So yeah.
Had we
Every-
known that before?
Good.
No, that's great to see. Thanks. I
Yeah.
don't always click on as many on
Yeah.
everything.
That's hidden. Yeah. We didn't mention it. We didn't mention it actually. Maybe mention it in one place, but yeah.
Well, you get people using stuff.
That's
They're
good.
just going to want more.
Yeah. That's great.
Are people coming up with more questions?
I was just going to ask in that demo you did, how much of that is scriptable? So I have, multiple structures I wanna step through and get the interactions out.
Um, so basically, if you can show everything in iCn3D, all the steps can be saved. So I think typically through iCn3D PNG image, because that works for files as well. But if your input is just PDB ID or AlphaFold ID, then the whole session can be saved in a shareable UI. Yeah, but in case, for example, if you've done so many steps and all the commands exceeded the threshold, which is about 4,000 characters, then only the iCn3D PNG image will work, not the
Right.
shareable.
No, I was thinking more the interactions. Say I have 10 structures, and I'd like to just export the interactions for all 10.
Okay.
Can we generalize the URLs, right? So because you're selecting, there's a selection step,
Yeah.
and then there's a interaction, step, right? Show interaction.
So in that case, I think it might be better to use the Node.js script. So in iCn3D Node, there are several examples for interactions. So I put ligand protein interactions. Okay, this is still old one, but you can show the protein interactions and detailed interaction, protein interactions. And so right now, for example, in the example, we are showing interaction for this structure between chain A and B. And here shows the, again, the peptide between two chains. In the ligand shows between the structure with this ligand, the ligand with all the rest.
Yeah.
But I think this can all be modified to define a set. For example, I just want to, I know which residue I want to study. For example, I want to study the interaction between a set of residues, for example, residue from 10 to 20 with all the mass interaction. I think you can also just modify the script to make that work.
Okay. With the caveat being, for any given structure, you don't know what the chains are named.
Right. Um,
Because there isn't a systematic chain naming, universal chain naming thing. Even if you look at immunoglobulins, sometimes it's H and L, which would make a lot of sense,
Right.
but it's not universal.
Right.
So be A and B in another structure.
So
I'm
one
asking
way,
these questions for Dylan.
Yeah, you're exactly right. And it sounds like Node.js is the way to go and not Python.
Well,
There
yeah,
seems to be way more flexibility with Node.js.
But
exactly.
what
Yeah.
Jiao's showing is there's a lot of Node.js scripts that have been written.
Yeah.
Right.
And so you can embed those in a script to automate it. You have to do that
That's right.
preliminary research for each structure of what you want to look at.
Right.
Yeah.
Right. Okay.
Yeah, go ahead.
Node.js script is very powerful, but it requires a little bit more, understanding of the code. So I just put all those example script there. But if you want to modify, you can contact me. I can help you to, achieve what you want.
Yeah.
Okay.
And for antibody, I think one way to tell, maybe you could use the IG domain detection if you are dealing with IG domain. But otherwise you can, get the change ahead of time from the structure and then define. Yeah.
Right.
Yeah. Mhm.
Other questions? Okay.
I have one. I have one.
Okay.
So can you do predictive folding? Like if you just have the translated amino acid sequence, put it in, and it will fold it into its three-dimensional structure for you then to use another known structure with a PDB file or another predictive structure to find out the interactions? Or do you have to have it already known by experimental data?
So we do have, this feature is kind of experimental. We cannot guarantee. So for example, we do have predict by sequence.
Mm.
And the ESM fold is very quick.
Oh, collab fold. Yeah.
Yeah. But, we are using their server. So it's a public server. It might be busy. But you could also make your own models, 3D models using AlphaFold too. So if it works, for example, it could be very efficient. So for example, I put any sequence. You put your sequence. I think it's limited to 400 residues. Then you have the structure right away. Usually it's very quick.
Cool.
Yeah. Then you can, for example, import your URL. So for example, retrieve by ID. So I want to, for example, any structure, I want append these. Then you have both structures in iCn3D, right? Both the your predict structures and your 3D structures. So you can do whatever you like with these both structures. And this can be saved. I think you can save as iCn3D PNG image and load back. So yeah.
Every year.
The AlphaFold, does that have a limit on it? I mean, because I played with the collab fold and it looks like you can do 20 a day.
Yeah, I don't know. Yeah, I think there's a limit. Yeah, probably. Yeah.
Probably. And is it 20? Is it 20 for the universe for iCn3D because it's going through NCBI, or is it
I didn't try.
20 for me?
I didn't realize you could do that in iCn3D. So I was doing it through SabDab and then bringing the PDB that I download from SabDab into iCn3D rather than doing it from within.
Yeah.
Um,
If it's a small protein like within 400, you can try the ESM fold. I think the
Okay.
quality also not bad. Yeah.
okay.
But your mileage may vary, right? One might be better
Yeah.
than another.
Yeah.
Yeah.
I think a really fun exercise is to take green fluorescent protein that's been solved and then have the structure predicted. Speaking of mileage varying.
Who can do it and make it make sense?
Yeah, which means like machine learning. So, you train on all these structures and then you say we have this accuracy against these structures. And then you put one of those structures in and you go, huh, what did you learn, machine? Or did the machine learn?
I don't know.
Cool.
Right? Yeah. So
Cool.
Are there other questions? I mean, it's just amazing to me that every month there's something new we can do that we couldn't do before.
Right. Right.
And it's already in iCn3D.
I mean, so we typically depends on the user request. So if you have any request, and if we can do it, then we might do it.
I'll put your email into the general section in Slack.
Uh-oh.
That suggestion.
Uh-oh.
I definitely think Erica's request, though, about a warning that you've tried to align a nucleotide sequence to a protein would be really good, because
Yeah.
my students do that too.
Yeah.
Lots of people do it. They just don't, one, it's just an accession number. So
Yeah.
what, right. And then, GAs, T's, and C's look a lot like A's and C's and T's and G's.
Yeah.
But then there's D's and V's and F's but they don't really look at those.
Well, and there are repetitive protein sequences that are like almost all glycine. So, I mean,
Yeah.
it's not
Right.
unheard of.
Right.
But the accession number should tell you, because NCBI should know that it comes from a nucleotide database and not a protein database.
Yeah, there are certain patterns. Yeah.
Yeah.
Should be able to ask about the origin. So, okay. Well, one more chance to ask questions, guys. All right, then. I'm going to stop the recording.